My name is Christine Flame. I'm working with the Alberta Agrisystems Living Lab, looking at KTT, that's knowledge transition transfer, as well as with producer engagement. And as part of that, we run a broad series of webinars throughout the winter season. So presumably you've already seen, but on our website, we have the links to all of those webinars. So for tonight, before we get started, I'd just like to acknowledge that the members of the Alberta Agrisystems Living Lab team are grateful to live, work, and be in relation with people from across many traditional territories, covering all regions of Alberta. This acknowledgement is to reaffirm our commitment and responsibility to improving relationships and our own understanding of local Indigenous people, their cultures, and knowledge. So with that said, I'm going to do a brief introduction to our speaker tonight, Dr. Breanne Tidemann, who's here with me. Dr. Tidemann is a research scientist with Agriculture and Agri-Food Canada in Lupins, Alberta. She grew up on a grain farm in West Central Saskatchewan, but completed all of her degrees at the University of Alberta. She holds a BSc in Biological Science and an MSc and a PhD in Plant Science with projects focused on weed science. Breanne started with Agriculture and Agri-Food Canada in 2016 as a weed scientist. Her research program in Lacombe focuses on integrated weed management strategies and novel weed management practices, as well as weed biology and ecology. And that's what we're going to learn about tonight. And just so everybody knows, if you have any questions, please feel free to use the chat or the Q&A. And at the end of the presentation, we will go through and make sure that we get each of those answered. So without any further ado, Breanne, why don't you go ahead and share your screen?
Good. Thank you, Christine, for the introduction. Now let's make sure I can actually see my screen all the way by just moving that out of the way. Excellent. Hi, everybody. Thanks for tuning in tonight. I really appreciate it. We are going through a whole bunch of weed science topics tonight, a whole bunch of weed science research. Before we jump in too far into the weeds, I just need to take a moment to first thank my team. I work with an amazing crew of 5 technicians. 4 of them have worked together for more than 27 years, so they are an incredible resource and incredible folks to work with. And then we always have an amazing group of summer students that we work with as well. I really do need to take the time to thank them. They're the ones that are actually on their hands and knees, noses to the ground, counting individual weed seedlings and all of those fun things that they get to do for weeds research. And I'm incredibly grateful for their time and their efforts. I also need to acknowledge the funders of my research program. So all of those funders that you see have funded various projects over the years, and we're incredibly grateful for their support and the fact that they give me the chance to study the things that I like to study. So very grateful to them. So what I was asked to do tonight is do a bit of a brief skim over quite a bit of sort of my program and even some of the other weed scientist programs on the prairies. So we're going to do a brief update on herbicide resistance and talk a little bit about the movement of kochia to new locations, because I think that's a really important shift that we're seeing right now. Then do sort of a skim across some of the current research that I've got going on or that's finishing up, as well as touch on some upcoming research that we're just kicking off. So very diverse, little bit higher level on most of these things. So if you have additional questions, absolutely feel free to put them in at the end of the presentation or to put them in throughout the presentation for answering at the end. But also feel free to follow up with me at any time if there's something that you're really interested in. Okay? So let's start by talking about herbicide resistance, everyone's least favorite thing to hear about if you're making weed management plans. So the first thing I just wanted to share is about the Prairie Weed Monitoring Network. So this is a new public-facing website where a lot of our prairie weeds information from the vast majority of the weed scientists on the prairies is going to start being collated. So you're going to find maps there from the weed survey and the resistance survey. You're going to find posters there. You're going to find fact sheets there, all kinds of things. And that QR code will take you to the website. So a big thank you again to all the funders on this project and something for you to check out. Hopefully I've given enough time for you to capture that with your phone. If not, I can always circle back to it at the end. Now, when we talk about resistance surveys, one of the last ones that was completed prairie-wide was 2014 to 2017, and that was led by Dr. Hugh Becky. And so Saskatchewan was done in 2014-15, Manitoba 16, and Alberta 2017. We've just recently completed the 2019 to the 2023 surveys. Those were led by Dr. Charles Geddes. Saskatchewan was done in 2019 and 2020, Manitoba in 22, and Alberta in 23. So when I say we just finished, literally Charles is still finishing up spraying a couple things. All of the major things were finished sort of early this year. So we did the survey in Alberta in 23, and it takes, a year and a half to 2 years to fully finish screening all of these plants. So it is a very large undertaking, and I'm very grateful that Charles leads that work. In addition, we'll touch a little bit on the kochia or the post-harvest surveys. Kochia gets sampled typically post-harvest because its seeds are not mature when we do the pre-harvest surveys. The ones that we're going to talk about tonight are from sort of 2018 to 2021. These are now being shifted and aligned a little bit more with the main herbicide resistance surveys, just so that it's not quite so extreme with surveying every single year, basically. So when we talk about unique herbicide-resistant biotypes in Canada, we sit somewhere around 86. And you can see our timeline is just on a fairly nice linear trend upwards. We see about 2 new biotypes reported each year. And again, really from, I'd say about 1990, late 1980s, we're on a pretty linear path upwards where we just continue to find more each and every year pretty much. So as I mentioned, the weed survey that we're going to focus on tonight was from 2019 to 2023. We visited a total of 827 fields, and we collected a total of 3,232 samples. Now, I am using the we obviously very royally. Charles led the study. I'm a collaborator on it, along with a number of other folks across all 3 of the prairie provinces. Now, this little graph on the top right shows sort of the percent of fields that had a herbicide-resistant weed in various rounds of the resistance surveys. And so what you can see is we are increasing, maybe not as quickly as we have in the past. A little bit about that is just we already developed some of the easy resistance to develop. And so the resistance we're developing now is a little bit more rare or a little bit more difficult to select for. So it does make sense that it is slowing down a little bit, but it's also about how much it's spreading. So there's sort of 2 factors there when we look at percent of fields. Now, when we actually did our sampling, I'm going to just focus on these top 2 numbers. 72% of fields where we collected a sample had a herbicide-resistant weed in it. So pretty high proportion there. Out of all 827 fields that we sampled, 70% of them had a herbicide-resistant weed in them. Okay. And what this comes down to is if you're dealing with a herbicide-resistant weed, you're either dealing with crop losses and yield loss because of the competition from those weeds, or you're dealing with increased costs to manage those weeds, whether that's using different herbicides or different weed management strategies. There's an additional cost. And so when we estimate the cost of herbicide-resistant weeds to prairie farmers, it's estimated that they cost $658 million per year for prairie farmers. So economically, also a massive concern. Now, these are the weeds that we most typically found and sampled. So in our broadleaf weeds, kochia was our most common broadleaf weed that we did sample for resistance testing, followed by lamb's quarters, wild buckwheat, Canada thistle, and spiny sow thistle. Our grass weeds, you're probably not surprised to see wild oat at the top, followed by green foxtail, barnyard grass, foxtail barley, and yellow foxtail. So again, those are what weeds we actually found pre-harvest that we were sampling. Now, if we look at the resistance profiles, these numbers are, so for wild oat, it was collected in 441 fields, and over 80% of those had a group 1 resistant wild oat in them. There wasn't enough seed in some of the populations to do all 441 fields tested to wild oat, but almost 50% of them had group 1 and 2 resistance. 435 fields here, again, about 50% had group 2 resistance. And you can see those broadleafs, quite a bit of group 2 resistance in various broadleafs. Now, each of these weeds is not found in every field. So if we take this away from the fields where they were collected to across all of those 827 fields, almost 50% of our fields had a group 1 resistant wild oat. About a quarter had group 1 and 2 resistance and group 2 resistance. And you can see the broadleaves are not as common. So where they're a problem, but they're not a widespread problem necessarily. Kochia, you'll notice, dropped to 30% of our fields. That is primarily because of its distribution, but we're going to talk about that a little bit further too. Focusing a little bit on herbicide-resistant wild oat, if you look across all 3 prairie provinces, we're looking at collections from about 450 fields, okay? 85% of fields where we collected wild oat had a Group 1 resistant wild oat, 50% had Group 2, 46% had Group 1 and 2. And we already talked about these numbers. We're about 50% of all fields and a quarter for the Group 2 and the 1+2. Really what I want to just kind of point out is if you look at the map, what we want to see is the little black dots. That means they were susceptible to our herbicides. And when you look at the map, there is just not very many black dots, unfortunately, okay? In Manitoba, there was actually no black dots. There was no susceptible wild oat collected for testing in this resistance survey, which is really unfortunate. Now, kochia. Kochia is a different beast that we're going to talk about. In most cases, we often don't bother testing for Group 2 resistance in kochia because we can't find Group 2 susceptible kochia anywhere anymore. In fact, we're often looking for Group 2 susceptible kochia for testing. So Group 2 resistant kochia is just considered the norm across the prairies at this point. If you have kochia, assume it's Group 2 resistant. In 2011, we had the first report of glyphosate-resistant kochia, or the first confirmation of glyphosate-resistant kochia. In 2012, there was a survey conducted to try and determine how widespread glyphosate-resistant kochia was, and it was about 5% of the populations that were surveyed. 5 years later, in 2017, we surveyed again, and our glyphosate resistance was up to about 50% of our populations, and we had also determined that we had some dicamba-resistant kochia showing up. Okay, so now we've got not just Group 2 resistance, not just glyphosate resistance, but we also have dicamba resistance, and we can have resistance to all 3 of those. Now, 10 years after it was initially found, so again, it was initially found in 2011, we surveyed again in 2021. Nearly 80% of our populations were glyphosate resistant. Okay, so it spread very rapidly in 10 years. Now, in addition to dicamba resistance, we've also determined that there is fluroxypyr resistance, another Group 4 product. Okay, and there can be dicamba resistant, fluroxypyr resistant, both of those, or either of those with glyphosate, or all 3. So again, multiple different combinations of what that resistance can look like. If we look at sort of where in Alberta this glyphosate-resistant kochia is found, you can see really all these counties have a pretty high proportion of glyphosate-resistant kochia. It is expected, essentially, at this point. If you're dealing with kochia, there's a very, very good chance that you're dealing with glyphosate resistance. Fluroxypyr-resistant kochia is not as common, although remember, this is in 2021. This is 4 years ago already. But you can see it was a little bit more almost on the western side. Unfortunately, we often have westerly winds, which will help it shift east because it likes to roll. So not as common as glyphosate resistance for sure, but it is still present in the populations. Dicamba-resistant kochia, you'll notice it's maybe less geographically aligned, but I don't know that that's a good thing because that also means it's kind of widespread in a sense. Again, less common than glyphosate resistance, but it is occurring in those populations. Now, unfortunately, in the last couple of years, we've also confirmed the presence of Group 14 resistant kochia in the prairies. It started out as 2 populations. Charles is continuing to screen, and he is continuing to find more and more populations of PPO inhibitor or Group 14 resistant kochia. Across the prairies, as I understand it, I believe he's up to about 18 populations now at this point. Don't quote me on that, but that's give or take. We're in the teens anyways. This was testing that was done with saflufenicil. However, he has found cross-resistance to our other common Group 14s with things like sulfentrazone in that group as well. So unfortunate, but we are seeing that Group 14 resistant kochia becoming more prevalent as well. What that means, if you have group 2, group 4, group 9, and group 14 resistant kochia, is your herbicide options become very limited. We get awfully reliant on group 6s and 27s in some of our crops. And if we are really completely reliant on them, it concerns me that in, 5-10 years or so, I might be talking to you about group 6 or 27 resistance. So something that we need to be cautious about, something that we need to be looking at other strategies for in addition to herbicides. Now, there has been this very common perception that kochia is a southern weed only. Okay, I work in Lacombe, I'm in central Alberta. A large number of my growers would say that kochia does not grow in Lacombe or too far north. And at this point, I am really quite forced to disagree. This picture of the train tracks in Lacombe was taken, I believe, 2 years ago, as was this gigantic kochia next to me. If you know me, you know I'm not super tall, but that's still a very big kochia. These pictures were taken in the city of Lacombe this year. Yes, that is all kochia in that bunch. Pretty much all of those little bushes that you see along the train tracks are in fact kochia. This is one of my students who kindly agreed to stand by the kochia for scale. Now, I know you're looking at it going, okay, but that's along the train tracks. It's in an urban center. These are 2 tumbleweeds that I chased across one of our research fields this spring, trying to stop them from spreading more seed. Okay, they are kochia. It is kochia tumbleweeds, and they are now rolling through our research fields, and I'm getting more and more reports from my producers in the county that they're seeing it in their fields as well. Okay, so this is spreading. It is not just a southern weed only. This is sort of, when we've done some modeling, this was done by Megan Vankowski and Charles Geddes, where they looked at sort of the niche or the habitat of kochia and sort of did a prediction of where it was growing, and it aligned pretty well with sort of what we thought of as kochia country. Now, they did some modeling looking at predicting it into the future, and it predicted this northward shift up into the Peace. And I remember seeing this about 5 to 10 years ago and being like, I don't know if we're really going to go that far north into the Peace. That seems very far north. This is a map from iNaturalist that I pulled just a couple weeks ago, where kochia has been found in the prairies. And so these findings in the Peace, these are findings up in Lacrete, with images that were sent in. And a lot of them, again, are more rural or urban finds. However, a lot of them are directly across the road from producer fields. So it's not going to take a lot. It takes 1 tumbleweed to roll into the producer's field and get things started, especially if you don't think it's there at all, because you're not watching for it then, and you might miss those first few seedlings getting established. This was from a producer north of Edmonton, who sent it to me and goes, is this what I think it is? And unfortunately, yeah, that is a great big patch of kochia, where there had been some disturbance, there had been some manure, so lots of nitrogen, and the kochia was just super happy to take advantage of that disturbance. So you'll notice I've been talking in the last couple of years in Lloydminster, in Wainwright, north of Edmonton, about kochia, because producers are actually dealing with it in their fields in those locations where, they've farmed there for 25, 30 years and had never seen kochia there in their lives. So even if you're a more northern producer, please take a look at some of the kochia pictures and know what it looks like, so you know what you're looking for, and hopefully you can stop it before it turns into a big outbreak. Because when they can produce 10,000 seeds plus per plant, it doesn't take very many plants to turn into an outbreak, unfortunately. 2 other weeds that I just wanted to talk about very quickly, water hemp and palmer amaranth, both pigweed species. Water hemp in particular has become fairly established in Manitoba. So this is the map from last year, from October of 2024, of all the counties that had water hemp found in it. Palmer amaranth has only been found in Manitoba so far once, in 2021. That population was eradicated, and they haven't found it since, but it has been there. Now, I bring this up just because these weeds are very big problems in their locations, which is more so in the US. However, they've been spreading. And again, they are pigweed species. We're talking 10,000 plus seeds per plant. most of those seeds that might get introduced anywhere are going to come with herbicide resistance. They're competitive weeds. They're really hard to control. So we don't want them, okay? The problem is they seem to be determined to make their way north. So water hemp and palmer have both been found in Ontario. They've both been found in Manitoba. Palmer was found in Washington. Both palmer and water hemp in Idaho, both in Montana and in North Dakota. We're kind of getting surrounded at this point, and I'm not really loving it, especially because weeds are not known for stopping to check their passports at the border. They just like to come right on over. The other thing with palmer and water hemp is their seeds can be moved by waterfowl, like geese and ducks. And so they will digest the seeds, and then when they pass them through, those weed seeds still have viability. I know you know how many geese there are in the fields around this time of year while they migrate, and so that's a little bit of a concern for me, is that there is potential introduction points. Last thing before I get off palmer and water hemp, if you're bringing any equipment, especially combines up from the US, please give them a really good solid clean in the US, because those seeds are very small. And if they come with the combine north, there's probably a few riding along in some of the nooks and crannies, and would be happy to establish a new population up here. Okay, so I'm going to move on from herbicide resistance, talk a little bit about some of the current research that I have going on. So I want to talk first about competitive varieties. So when we talk about competitiveness, usually we kind of break it into two categories, a focus on crop type and then a focus on crop variety. Now, there's quite a bit of information available if you dig back through the research in terms of information on which crop types are more competitive than another. So if you go back through a bunch of them, typically you'll see barley is one of the most competitive, then canola. Sometimes those are equal to each other, which is better than wheat, which is better than flax, which is better than soybean, which is better than most of the pulse crops. That's kind of the hierarchy. Now, yes, environment plays a role. Yes, your agronomy plays a role, but that's generally sort of how they compare. Now, for years at this point, I have been talking about that it's also beneficial to use competitive varieties. And the most common question that I would get is, where can I find that information? And I would go, well, you can't. That's not really information that's available. And I started irritating myself to the point that I really wanted to dig into this a little bit more and see, can we actually rank varieties based on their competitiveness? Now, we know there are characteristics that are generally associated with competitiveness, things like height, early season vigor, the speed of emergence, the biomass they produce, things like that. But you're really going to have to dig and be digging in the literature or in different variety trials, I guess, to try and find those traits and be able to compare them between varieties. So again, not easily accessible information. And so what we wanted to do is look at wheat and barley varieties in separate experiments. So we're not comparing the wheat to the barley, we're comparing wheat to wheat and barley to barley. And we wanted to look at the ability to compete, which means that variety actually is reducing the weed biomass, or there's less weed growth, and the ability to withstand competition, which means even when there are weeds there, the variety is yielding pretty well. Now, we also wanted to look at some different classes, so I wanted to make sure I had some wheat varieties from CWRS and CNHR as an example. In barley, I wanted to make sure I had some malt varieties and some feed varieties. And the question really was, can we rank varieties for competitiveness? But also, when we're doing these rankings, is this something that we really need a weed community, we need multiple weed species in, or can we just use a model weed for simplicity, for ease, for maybe fewer locations being willing to do it if they don't have to seed a bunch of weeds down to do the research type of thing? So we did this at 4 sites. We had 2 in Alberta, Lethbridge, where Charles Geddes was the site lead, and Lacombe, where I was. And then we did 2 in Saskatoon, one at the U of S with Chris Wellenberg and one at Ag Canada Saskatoon with Sean Sharp. Now, my 2 in Saskatoon, Sean, Charles, and I all set up our sites where we had weed communities there. So we had regional weeds that were specific to our area. So I had wild oat, I had volunteer canola, I had cleavers, whereas Charles had wild oat and kochia primarily. Okay, so regionally specific to try and capture that variation. The U of S site only used volunteer canola as their single model weed, a single pseudo weed, to see if their rankings would match up with Sean's. Not identical locations, but close, similar precipitation at least. And so we would expect to see similarities in their rankings. Okay. So this is how we set up our trials. Each pair of these is a single rep. Okay. Now each rep is broken into two. This is our weed-free or relatively weed-free. You'll notice most of those weeds are in the interplots, and the back is the weedy in this case. In this rep, the weedy's in the front, and the weed-free is in the back. The same variety goes across this way. Okay, so the same variety is just separated by the little gap in between. And then we have the weedy and the weed-free. And so that's where we were looking at sort of our weed biomass, but also our yields as a percent of when there was no weeds there and those kinds of things. So that's what our trial setup looked like. These are the varieties. Now, I'm not focused so much on these specific varieties. It was more, can I pull them apart? Than actually developing a full-on ranking from this, if that makes sense. It was a little bit protocol and idea development more so than these specific varieties to give the information. So when I'm showing you where things land, please don't take that to the bank as that is the final competitiveness being given to that variety, because there is still more work to do on this, okay? So you'll notice that we have 6 CWRS varieties. We've got 4 CPS, CNHR CWSP, CWSWS, and CWHW. The proportion or the number of varieties was really based essentially on crop insurance acres. So CWRS had the most acres, it got the most varieties. CPS had the next most acres, it got the next most varieties. Okay. Barley, we had our 5 malts, our 4 6-row feed forage varieties, and then our 4 2-row feed varieties. Okay. Again, I'm not going to focus specifically on the varieties through this talk. Kind of data we were taking emergence dates and counts. We were looking at ground cover. We were looking at leaf width, crop biomass, date of canopy closure, crop height, lodging, maturity, yield, 1,000 kernel weight, dockage. So we are going to try and link some of those traits again to sort of our competitiveness ranking. And then also just looking at that biomass and that yield as sort of our 2 main components. Now, I said I'm focused on varieties here, but I had to take a moment to just show you some crop type things. Because of how we had this set up, we always had our wheat and our barley trials side by side. So this trial here is our barley trial. You'll see the weedy in the back, then the front. And it has weeds in the weedy, but it's fairly clean. This is the wheat. You will notice just a couple more weeds in the weedy on the wheat side. Okay. These were seeded within a day of each other. The weeds were put down the same way at the same rate between the two. I've had comments that, well, it's because these pathways are sprayed out and those ones are not. So I went so far as to cover those up completely, and there's still a lot more weeds in the wheat. Okay, so I think it's safe to say, even just visually here, that yeah, barley is a little more competitive than wheat generally. So if you're really dealing with a weedy field, barley might be the better cereal choice in that field. Can't back the data up, but again, maybe here a picture is worth a few stats anyways. Also, along those lines, just the weed size. So here's our barley, one of our barley plots on the left. Yes, there's some weeds in there, but the lamb's quarters in the barley is a little smaller than the lamb's quarters and the canola in the wheat. So again, that crop date did seem to be a valid recommendation still. Some of those weeds were so large that we actually took our crew picture, and we've been complimented on sort of the trees providing a very unique background. Unfortunately, those are all lamb's quarters from the wheat trial. So they were a little large, safe to say. But if we focus in on the competitive varieties, which is what we're supposed to be talking about, this is a fancy looking graph for barley. So I'm going to break this down a little bit. In this corner, this top right corner, these are the varieties that had the lowest weed biomass and the highest yield, even in our weedy conditions. Okay, the top left and the bottom right either had low biomass but didn't yield as well, or they yielded pretty good but they had a higher level of biomass. And then our bottom left was high weed biomass and a low yield. So those were our low competitive varieties. So top right corner was highly competitive, bottom left was our low competitive. And the other two were intermediate is what I'm calling them. Now the dashed lines is the averages across all the varieties and in this case across the locations. So that bar will shift each year and with each season. It's a relative comparison, basically, is what we're doing. Same way we do relative yield and relative maturity when we do our variety trials. Okay, so with the barley, yes, we can pull these apart. We can separate them out. We can actually just by plotting these values, put them into 4 different quadrants of competitiveness. Similar story for the wheat. We can pull this apart. We can get them into different quadrants. Some are more competitive than others. Some of them have a lot of wheat biomass, but still yield decent. Okay, so they shift in terms of where they fit, basically. But there is also quite a bit of variability. You see these error bars and you see they are quite large. So there is variability. Again, this is across years and across locations. So a fair bit of variability is not surprising. Now, one of the things that does concern me with this idea is the consistency that these varieties land in different categories. So I'm going to use AAC Iceburn here as an example. It was highly competitive across locations and at every individual location. So it seems to be a fairly highly competitive variety relative to the other ones that we tested. That was great. However, AAC Brandon here was, again, fairly consistent, low competitive, maybe an intermediate competitive in Lethbridge, but again, generally fairly predictable. AC Andrew, though, was low competitive across locations, intermediate at Lacombe, highly competitive in Lethbridge, and low competitive in Saskatoon, which to me suggests perhaps there is some regionality to some of these varieties. And that may be crop class, or it could be variety specific. There are some varieties that are probably just better suited to different regions, which means that if we are wanting to do this type of competitiveness ranking, it probably needs to be done regionally with something like the regional variety trials, which means a fairly large investment to be able to do this in all of those different regions to make the information useful to the producers. So that's a little bit of concern for me is there's a significant amount of regionality here. In terms of one weed or a weed community, the weed community seemed to be the better option. You can see in between Saskatoon and U of S, we have some very different rankings, and I would lean more towards the weed community as being representative than a single pseudo weed based on what we've seen. So next steps with this type of research, a big question to me is how does it compare to seeding rate or other integrated weed management strategies? So does it pay for me to go from a low competitive variety to a high competitive variety? Or should I just increase my seeding rate of the low competitive variety and I'll just match what the highly competitive variety can do? Because if I can just increase my seeding rate, do we need to make that big infrastructural investment into actually determining competitiveness in the different regions? Is it worth it, or should we just bump seeding rates? Okay, so that's an interest to me. Again, the investment or the infrastructure would be needed to categorize all varieties regionally if this is something that we are going to pursue. And so it takes an investment, and it would take protocol development and getting collaborators on board and things like that. So is it worth it? And does it have a big enough impact to warrant that investment? So more research to do, basically. We're going to talk a little bit about lupins. So we're changing projects here, talking about lupins. Why are we talking about lupins? Well, there's been interest in lupins a few times as a potential crop in Alberta. If you go back in the literature, if you go back to people that have worked into the industry, they're going to be like, how didn't we talk about this 10, 15, 20 years ago? Yes, we probably did. Right now, there's interest in lupins because they are resistant to Aphanomyces root rot. Okay, so that is why there's a renewed interest in lupins is, hey, maybe we can bring this in on some of the pea acres that are being affected by Aphanomyces. A challenge is they are poor competitors like other pulses. So they're a pulse crop. They're not great competitors. When we are bringing in a crop like lupin, Unfortunately, the vast majority of our herbicides are not registered on Lupin, or if they do happen to have Lupin on the registration, a lot of those products are Eastern Canada products, which is great. It's fine. They're Canadian registrations. However, the only information we have is how well it works on Eastern Canadian weeds, things like ragweed or fleabane, that are not really helpful when we're dealing with kochia and wild oat and cleavers. Okay, so we're missing some information there. So what we did is in 3 locations over 3 years, sort of in a pocket where I was told lupins would be best fit, we tried a bunch of different herbicides, and I'll show you those next. And we used, again, common regional weeds at each location. And we looked at both crop safety and weed control efficacy. So these are the treatments. Now, I do have all active ingredients here. I apologize. As a weed scientist, I primarily work in active ingredients because the trade name changes depending on who I'm talking to and where they're from. So some of these you're going to recognize. Some of them, again, are more Eastern Canada weeds. So we were looking at sulfentrazone and pyroxasulfone separately, and then the combination of the two. Flumioxazin plus pyroxasulfone, lorazolam, saflufenyl, tepramazone, pyroflufen plus MCPA or plus bromoxynil, pomazone, dimethenamide P, which does require HARO incorporation, so we did do that, and S-metolachlor, again with HARO incorporation, and fomesafen. All of these 14 treatments are pre-products, so they're sprayed preceding or pre-emergence. They're soil applied and they have residual. I shouldn't have said soil applied. There are some that are more of a burn down type treatment, something like, say, saflufenyl. That is more of a burn down treatment and less of a soil residual. So we are kind of looking at things that we could potentially layer with, things that we could potentially add as a pre-seed burn off as well in that list of 14. The second group over here, tepramazone, clethodim, fomesafen, diflufenacin, and metribuzin, these were sprayed post, so these were sprayed in crop. Yes, there is a repeat of tepramazone and fomesafen. We tried them both pre and post. And then we had 2 controls. We had a weedy control, untreated, let the weeds just grow, and then a weed-free where our wonderful students got to hand weed or hand hoe those treatments and keep it weed-free so that we had a true weed-free yield to compare to. I'm not going to go through every weed biomass and every rating and everything like that. I'm just going to give you a couple takeaways. In terms of crop injury, we had a couple products, post, tepramazone and fomesafen, that did a number on our lupins. So much so that one of them, within the first couple days after we sprayed, one of my technicians came and asked me if they were supposed to be fully dead, which they were not, and they weren't. They did actually come back, but there was a hit to yield and a hit to biomass. So there was definitely some crop injury there. Our pre-products, we saw very limited injury. We had a little bit of injury at some locations that might warrant some additional study. However, I do asterisk our crop injury results because with all of those soil applied residuals, you have more activity when you have precipitation. Now, I swear it's because we were working with soil applied residuals, but the 3 years we did the study, we had dry springs. So we didn't see crop injury, but we also didn't necessarily have the precipitation to see crop injury. So they look really good now, but I would really like to see them in a wet spring to actually confirm that, they do have good crop safety. Or some additional controlled work in a greenhouse or a growth chamber where you keep them really wet to confirm that crop safety. Weed control, it obviously really depended on the product. So if we're spraying clethodim post, well, clethodim's only going to work on my grasses, right? So it worked really well on wild oat, didn't work so well on volunteer canola or cleavers. However, across the study, one thing that really stood out to me is that volunteer canola is going to be an issue in lupin. Okay, we had a heck of a time controlling volunteer canola without also controlling the lupin in terms of severely injuring it. So weed control of volunteer canola is going to be a challenge, and something that we're going to have to really think about how we manage that appropriately if lupin are actually going to have a place as a crop in Western Canada. Final takeaways on this: more work needed to ensure crop safety with the pre's. Like I said, we need rain. to really test that. I really think from what we saw, there was no single obvious, like, oh, this product is the best fit for lupin. I think it's going to take a layering strategy. So combining a pre and a post um, with some residual and a burn down product, I think it's going to take a little bit more complex of a strategy, and maybe some mixtures. Metribuzin is sort of the standard right now for use in lupins, and it looks okay. However, as far as I can tell, there's not graminicide tank mixes on the metribuzin label, and you kind of need both aspects of that. So I need to check further into that, but that's a little bit of a concern with me. Um, Really, what I'm seeing is we're going to have to look at probably some other integrated strategies as well. I don't know if lupinar may be a good option for paired row just to better spread out their crop competition. Possibly some work on seeding rate in combination with the herbicide layering and the herbicide mixture. So I think it's going to be a fairly strategic management strategy on these ones. But the other thing is rotational sequencing will be important. If volunteer canola is really hard to control in lupin, we maybe don't want canola as the crop prior to lupin, which unfortunately is kind of where that would probably fall in most rotations. So thinking about that rotational sequencing and what weeds are going to come through from that prior crop, it's also going to be important. Drones and herbicides. I'm going to go through this fairly quickly. There's some work that's been happening looking at hyperspectral drone imaging and kochia resistance. And so this started in a greenhouse setting with a hyperspectral camera, moved into plots with the drone in the field. And they're actually able to predict resistance to glyphosate and fluroxypyr quite well, even before the plants are actually sprayed. Okay, so this is using the images and machine learning and AI. The model was trained on the greenhouse data first, but you can see with glyphosate, even before spraying, it was predicting resistance with about 74-75% accuracy. This one actually up to 90-some% in the field, even prior to spraying. Fluroxypyr not quite as high, but in the mid 80s. So actually fairly impressive considering it's before it was sprayed. Now, I'm going to say right here and now, I am not a digital ag specialist. I was the weed science herbicide part of this, and I'm going to show you the scientist that was the drone AI machine learning side of this. If you have questions on the drones and the AI and that side, I'm going to point you to him, because again, I was good at the weeds, and I nodded and smiled a fair bit when he was telling me about the drone and the machine algorithm side of things a little bit. Now, we did something similar on wild oat, but we didn't do the greenhouse component first. We just took it to the field. And we did wild oats and peas with a group 2. We did wild oat in wheat plots with a group 1 herbicide. This is my colleague Kashav, who's the scientist from Lethbridge, flying his drone and doing some data acquisition. And again, with our group 1s, we're actually sort of up in that high 80s already for prediction and comparison to our visual ratings and our ratings of resistance. And with the group 2s, a little bit less so, but still impressively high in my opinion. Higher than I expected it to be, if I'm being honest. We've also done some work looking at imaging with the drones to predict lentil desiccation timing. So this is comparing visual ratings to sort of what it was picking up with the drones. And so the visual ratings are quite variable between our 2 locations. However, moisture content did a whole lot better across locations. So the idea here is that it's sometimes hard to tell with the staging and variability across a field if you're at desiccation timing or not. So maybe we can go bigger, maybe we can do some drone imaging and get an average moisture across the field, because the drone can get that much higher view than sometimes we can rate from ground level, because we don't want to go through the entire field and sample. So that's what we're working towards. We've obviously still got work to do. Like, these are not high enough to be going field scale yet. But some interesting ideas here in terms of how to use that imagery to help us deal with some of our weed challenges. So as I mentioned, I am not a digital imaging research specialist. That would be Dr. Kashav Singh from the Lethbridge Research and Development Center, and he is a specialist in digital agriculture and knows far more about the technicalities of all this research that I've just showed you. If you want to know what we sprayed or what we used to spray it or where I got my wild oat populations from, I'm your girl. If you want to know about the drones and the algorithms, Kashav's your guy. Okay. So that's kind of my very brief skim across that little section of research. I wanted to talk briefly about harvest weed seed control and wild oat distribution. I love getting to slide harvest weed seed control into my talks. I've been working on this for over 10 years at this point, and it's kind of a pet topic. When we talk about wild oats and harvest weed seed control, so this is things like the seed destructors and the impact mills, okay? We know wild oat drop a lot of seed prior to harvest timing. And so I have typically been saying that they don't make the best target for the impact mills. However, even those few seeds that remain can be spread more than 250 meters across the field by our combines. And so particularly if those seeds that are remaining are from resistant wild oat populations, we don't really want those spreading further across the field. We'd rather keep those in a patch where maybe we can do some patch management or deal with it in this area rather than the entire field. So the question that we asked was, can you keep a wild oat patch a patch by using harvest weed seed control? So using an impact mill in this case. So we've actually been studying this in a producer's field in Saskatchewan. We've got just finished off our 4th harvest, and I don't have that data in here yet. But this was patch sizes either in the no-mill fields or in the mill fields. So in the no-mill fields, my producer is actually turning off his impact mill for us, and we're able to look at sort of the change in patch size. Now, just by chance, they were randomly assigned. The no-mill fields were a little bit bigger patch area right off the bat, but you can see they seem to be increasing in size faster than the milled patches. Now, again, we just did another harvest this year. I got to get the 2025 data in here yet, but so far it is looking like it can help us at least keep those resistant weeds in one location rather than making it an entire field problem. And this is something that I have heard from a few producers that are using the mills, that they've actually seen this quite visually with kochia, where they were getting busy. The combine with the mill wasn't there to do the kochia patch area, and they just said, to heck with it, we're fighting weather, take the other combine through. And they said where we chose to do that, the kochia has moved significantly larger in our field. It's not our nice small patch that it once was. Whereas in the fields where they used the mill in the kochia patches, it was in the same area that it was the year before. So they said we didn't notice a massive change in density from just a year or two of using our mill, but we really noticed where we didn't use the mill in terms of how much bigger that patch had got. So that was, again, a really brief skim, and that's not all the research that I've got going on, but just some that I thought might be of interest to the group. I want to just briefly talk about 2 upcoming research projects that I've got going on, one on weeds and pulse diseases and one on directed energy. So directed energy, it's actually a patented combination of energy forms. It's looking at blue wavelength light and infrared wavelength light. And the idea is that this will be developed into a new form of harvest weed seed control. So instead of using a mill, which is high energy and high upfront cost, the idea is that you could use this directed energy combination for smaller combines, using less horsepower and at a lower cost is the goal. So the goal is sort of to target some of those older combines that maybe don't have the horsepower to actually drive some of those impact mills. We have a benchtop version of this unit, and we're going to be testing efficacy on prairie weeds. There's been some work done in the US where they are finding decent efficacy with this combination of energy forms. And so we're going to be testing it on wild oats, kochia, volunteer canola, cleavers, green foxtail, all of those big weeds, wild buckwheat. We're going to be running through this unit and seeing what these combinations of energy does to them in terms of their viability. Now, there is not yet a commercially available unit for harvest weed seed control for combines using directed energy, but they are in development. Okay, so this is something that we do expect to see coming to market. And so we want to be able to provide some information on what weed species does it work on, how well does it work, some of those kinds of questions. So that's a pretty cool project that we will be starting in the next month or so, actually. We just got our unit this fall, and we had to get some cord ends wired up and find the right voltage of power and things like that on the station to set everything up. And we'll be, right now, my students are having all kinds of fun counting weed seeds. They're counting to 100 many, many, many times. Weeds and pulse diseases. So I have a master's student working on this, Logan Snow. He's a University of Alberta master's student. And we've kind of started working on this because of black medic. So black medic, probably a weed you know from your lawns, and probably hate a little bit. It is becoming a significant issue, particularly in Saskatchewan, in lentil growing areas. Okay, so it's becoming a weed that if you have it, you really kind of hate this one, and are having some trouble controlling it. Now, one of the questions that I've had is black medic is a leguminous weed. Okay, it is actually essentially in the same family as peas and lentils. And so the question is, can it also be infected by Aphanomyces? So one of our main recommendations right now dealing with Aphanomyces is crop rotation. Rotate away from susceptible crops. Well, what happens if our weeds are also susceptible and we're having trouble managing them? Are they just making the Aphanomyces issue worse? Okay, so black medic in particular is an interest. This is sort of analogous to me to clubroot and brassica weeds. So if you have shepherd's purse, if you've got stinkweed, if you've got volunteer canola, and you purposely rotate it out of canola to try and get your clubroot spore levels under control, those weeds can still be continuing that clubroot cycle, right? We're asking, can black medic do the same thing? So that's what we want to look at. We also want to look at the fusarium root rot, so fusarium avinaceum in particular, and looking at how it can infect common weeds and how they may be contributing to the inoculum as well. So Logan just got started this September. This is a study that's just underway. We did do some field survey collections this summer, and we'll have a whole bunch more to go next summer. But trying to dig into this and just see what role black medic might be playing that we didn't really expect it to be playing now that we're dealing with Aphanomyces on the prairies. So massive amounts of information, massive different areas and different research projects. I hope I didn't completely overwhelm you with anything. I hope you got one or two things that were useful out of that. If I completely bombarded you and you're like, I have, a few notes about things and I need to follow up with you, I'm very available online. I'm on pretty much every social media. So I'm on X, I'm on Instagram, I'm on LinkedIn, I'm on Blue Sky. My email address is here. If you Google me, you can probably find my phone number pretty easy if we're being realistic. Feel free to reach out at any time. I'm happy to chat. And I did see a few questions coming in, so I'm happy to start having a chat about some of those as well. Thank you for taking the time tonight.
Wow. Thank you, Breanne. That was great. There's a lot more going on in the weed world than I was aware of. Yeah, we'll take a look at a couple of the questions that were posted in the chat. Earlier on, could you give an example of herbicide active ingredient in group 1 versus 2?
Absolutely. So our group 1s are our FOP stem stems. So cladinofop, clethodim, pinoxidin. Those are all group 1 herbicides. Group 2s would be things like your IMIs, your imazethapyr or mazamox, or something like, I'm blanking, flurazolam. Simplicity has group 2s in it. Everest is a group 2, things like that.
Great. There was a couple comments here as well about seeing kochia spreading.
Yes.
You were saying.
I've actually just been asked to do another couple of talks north of Edmonton or in the Edmonton Highway 16 corridor.
Yeah.
It is definitely moving in that region and not just urban, but also in producer fields.
Yep. Yes. A little bit further down there, does the variety X weed competitiveness have any linkage to the breeding program location?
So not that I've seen as of yet, but I am still digging into that a little bit. We're still digging into the classes. It doesn't appear to be a big driver right now. Breeding programs didn't seem to be a big driver, but we're still looking at some of those correlations and seeing if there's factors there that we can pull out.
And could you maybe describe the mill that you were talking
Yeah.
about? I was not
Yes.
familiar with that either, so this would be great.
Yes. I will try not to get on too much of a soapbox. If I do, someone flag me down or put stop in the chat, okay? So harvest weed seed control, the idea behind harvest weed seed control is that most of our weed seeds are probably coming out of the combine in the chaff, okay? Just based on how the combine separates. We might have some go into the grain tank, might have some go out with the straw, but the majority will come out in the chaff. And so harvest weed seed control is a whole paradigm of potential control ideas that focuses on managing the chaff. So the one that we think is most suitable right now for Western Canada is impact mills. So if you've heard of Harrington seed destructors, seed terminators, seed control units, these are all commercial versions of impact mills, is what we classify them as a group. So the idea of an impact mill, it's based on the idea of like cage mills or hammer mills that were used in mining and gravel crushing or coal crushing at one point. But the idea is that basically they're spinning really fast, and the chaff is going into the middle and has to go through those spinning cages or spinning panels, I guess, if you will, to get out. And so it's going to get hit multiple times, and it breaks it down into smaller pieces. And when those weed seeds get broken down into itty bitty little pieces, they can't germinate anymore. So it destroys their viability. Ray Harrington, who was the original inventor of the Harrington seed destructor from Australia, he described it as picture yourself standing on one side of a double lane highway at rush hour on a long weekend, and you're only allowed to walk across the lanes and no one's stopping for you. Chances are you're going to get hit multiple times in multiple directions and probably not be alive by the time you get out of there. Same idea is what we're trying to do to the weed seeds. So we've done some stationary work. Really, anything that goes into the mills seems to be controlled really well, like 98% plus control of the seeds that are going in. Really high control. The problem is getting it from the plant into the combine. So if this plant is already dropping the seeds to the ground before the combine comes in, or if the combine knocks a lot of those seeds off, then we're not getting it into the combine. We can't actually get it into the mill. And so that's been a challenge with wild oat, because wild oat likes to drop its seeds early. It's hard to actually get them into the combine. But the question was, even if we're only getting 20 or 30%, at least we're not spreading them, because wild oat likes to just drop its seeds beside its plant. It's not like kochia, where kochia is going to go for a roll and spread its seed no matter what we do, in a sense. Wild oat likes to stay where it is, except for when we willingly go, here, go see the rest of the field, go visit with the combine. So that's why we were focused on wild oat in that particular study. Is it doesn't really like to go anywhere anyways, but we do help it. Can we just keep it here instead? So that's what the mills are. Hopefully that helped.
Interesting.
Yeah.
Well, thanks everyone for your questions. If there was anybody else that wanted to put their hand up or ask a question directly, you could do so now. But otherwise, I mean, we're right at the end of our time. So thank you very much, Breanne, for that excellent presentation. I learned a lot.
No problem at all. And seriously, I'm happiest when I get to talk to people that are interested in weeds and some of the research. So if there are any questions or any follow-ups, feel free to reach out to me at any time. It's like, yay, someone cares.
Yes, somebody does care, thankfully.
Yes.
We need the research. Thank you so much for your time. And thank you everyone as well for joining us this evening. So
just a question that came in here. Any trials with weed zappers? I have not got to play with the weed zappers yet, which is electrical weeding, which typically gets done over top of the crop. So you need a separation between the crop and the weed, or it's non-selective and is being done right on the ground. So think burn-off type treatments. I haven't done any work on it. From what I've seen in other regions, it can work fairly well on broadleaf weeds, does not work as well on grasses. So grasses, you'll hit the main stem, but you won't get the tillers, is what I've seen. Unfortunately, a lot of my colleagues have only been looking at the kill and not at the seed viability. So there's things that I would like to do and things that I am twisting colleagues' arms to get them to do for me because I haven't got my hands on a zapper yet. If any of you have a zapper on your farm and would like to let me come play, please reach out.
Sounds good. Eric, you raised your hand. Do you have a question, Eric? Please. We just put it in the chat. I think you should be able to speak. Oh, here. There. Okay. Eric, if you want to unmute yourself, you could ask a question.
Okay, there we go. About 40 years ago, I was at a Charlie breeder's farm just west of Dawson Creek, and he was talking about his wild oats. There weren't a lot of herbicides being used in this is the late 60s, early 70s. But what he would do is plant sweet clover, and we had a field that had too many wild oats, and he'd take that off his hay that summer, and then the next year he would take off the sweet clover, either combine it or bale it up again. So he had 2 years without the wild oats being able to go to seed, and that was organic. Long before that term showed up, but that was one of the ways it was being done. It's maybe not that easy for straight grain farmers, but it sure works well with a mixed farm.
Yeah, perennials for sure. We've done some work with alfalfa as well. That can be great. 2-3 years of cutting it, fantastic because you're preventing that seed set. Even early cut barley silage can be phenomenal for stopping wild oat seed set. But again, if you don't have a use for that feed, that's where the challenge comes in for the grain farmers.
Yeah, great comment. Thank you. Okay, well, if there is nobody else, we'll say goodnight. And thank you, Dr. Tidemann, for joining us this evening.
No problem. Thanks all for tuning in.
Yeah, thank you, everybody, for coming.
Good.